animal systems in which the germline is set aside during early embryogenesis, the germline in plants is established only after the transition from vegetative to reproductive development, that is, flowering. The vegetative and reproductive structures of the shoot are all derived from the shoot meristem formed during embryogenesis. Clonal analysis indicates that no cells are set aside in the shoot meristem of the embryo to be used solely in the creation of reproductive structures. Irradiating seeds causes changes in the pigmentation of some cells. These seeds give rise to plants that have visually distinguishable sectors descended from the mutant cells. Sectors may extend from the vegetative portion of the plant into the reproductive regions, indicating that maize embryos do not have distinct reproductive compartments. Maximal reproductive success depends on the timing of flowering and on balancing the number of seeds produced with resources allocated to individual seeds. As in animals, different strategies work best for different organisms in different environments. There is a great diversity of difference among the over 300,000 angiosperm species, yet there appears to be an underlying evolutionary conservation of flowering genes and common patterns of flowering regulation. A simplistic explanation of the flowering process is that a signal from the leaves moves to the shoot apex and induces flowering. In some species, this flowering signal is a response to environmental conditions. Fundamental pathways leading to flowering are regulated at numerous control points in different plant organs, roots, cotyledons, leaves, and shoot apices in various species, resulting in a diversity of flowering times and reproductive architectures. The nature of the flowering signal, however, remains unknown. Some plants, especially woody perennials, go through a juvenile phase, during which the plant cannot produce reproductive structures even if all the appropriate environmental signals are present. The transition from the juvenile to the adult stage may require the acquisition of competence by the leaves or meristem to respond to an internal or external signal. Graft organ for experiments, mutant analyses, and molecular analyses give us a framework for describing the reproductive transition in plants. Figure. Experiments have identified the sources of signals that promote or inhibit flowering and have provided information on the developmental acquisition of meristem competence to respond to these signals. Analyses of mutants and molecular characterization of genes are yielding information on the mechanics of these signal response mechanisms. Leaves produce a graft-transmissible substance that induces flowering. In some species, this signal is produced only under specific photoperiods, day lengths, while other species are day neutral and will flower under any photoperiod. Cells may be competent to perceive or pass on photoperiodic signals. The phytochrome pigments transduce these signals from the external environment. Structure of phytochrome is modified by red and far light, and these changes can initiate a cascade of events leading to the production of either floral promoter. Institute for Advanced Studies, Jodhpur 67. Or floral inhibitor. Leaves, cotyledons, and roots have been identified as sources of floral inhibitors in some species. A critical balance between inhibitor and promoter is needed for the reproductive transition. In some species, meristems change in their competence to respond to flowering signals during development. Vernalization, a period of chilling, can enhance the competence of shoots and leaves to perceive or produce a flowering signal. The reproductive transition depends on both meristem competence and signal strength. Figure. Culture experiments in several species, including tobacco, sunflower, and peas, have demonstrated that determination for reproductive function can occur before reproductive morphogenesis. Isolated shoot tips that are determined for reproductive development but are morphologically vegetative will produce the same number of nodes before flowering in situ and in culture. The black box between environmental signals and the production of a flower is vanishing rapidly, especially in the model plant Arabidopsis. The signaling pathways from light via different phytochromes to key flowering genes are being elucidated. Molecular explanations are revealing redundant pathways that ensure that flowering will occur. Light-dependent, gibberellin-dependent, vernalization-dependent, and autonomous pathways that regulate the floral transition have been genetically dissected. Ancestral angiosperm is believed to have formed a terminal flower directly from the terminal shoot apex. In modern angiosperms, a variety of flowering patterns exist in which the terminal shoot apex is indeterminate, but axillary buds produce flowers. This is observed as an intermediate step into the reproductive process, the transition of a vegetative meristem to an inflorescence meristem, which initiates axillary meristems that can produce floral organs, but does not directly produce floral part cell. The inflorescence is the reproductive backbone stem that displays the flowers. The inflorescence meristem probably arises through the action of a gene that suppresses terminal flower formation. The CENTRORADIALUS CEN gene in snapdragon suppresses terminal flower formation. It suppresses expression of FLORICAULA flow, which specifies floral meristem identity. Curiously, the expression of flow is necessary for CEN to be turned on. This homologue of CEN, terminal flower 1 or TFL1, is expressed during the vegetative phase of development as well, and has the additional function of delaying the commitment to inflorescence development. Overexpression of TFL1 in transgenic Arabidopsis extends the time before a terminal flower forms. TFL1 must delay the reproductive transition. Garden peas branch one more time than snapdragons do before forming the flower. That is, the axillary meristem does not directly produce a flower, but acts as an inflorescence meristem that initiates floral meristems. Genes, DED and VEG1, are responsible for this more complex inflorescence, and only when both are non-functional is a terminal flower formed. Figure. The next step in the reproductive process is the specification of floral meristems, those meristems that will actually produce flowers. Arabidopsis, leafy LFY, Apitella1, AP1, and cauliflower cal are floral meristem identity genes. LY is the homologue of flow in snapdragons, and its upregulation during development is key to the transition to reproductive development. Expression of these genes is necessary for the transition from an inflorescence. Institute for Advanced Studies, Jodhpur 68. Meristem to a floral meristem. Mutants LFY form leafy shoots in the axils where flowers form in wild-type plants. They are unable to make the transition to floral development. If LFY is overexpressed, flowering occurs early. For example, when aspen was transformed with an LFY gene that was expressed throughout the plant, the time to flowering was dramatically shortened from years to months. AP1 and CAL are closely related and redundant gene. The cal mutant looks like the wild-type plant, but AP1 cal double mutants produce inflorescences that look like cauliflower heads. identity genes initiate a cascade of gene expression that turns on region-specifying catastrophe genes, which further specify pattern by initiating transcription of floral organ identity genes. Weigel 1995. Superman sub is an example of a catastrophe gene Arabidopsis that plays a role in specifying boundaries for organ identity gene expression. 3 classes A, B, and C of organ identity genes are necessary to specify the 4 whorls of floral organs figures. They are homeons but not Hox genes and include AP2, AGAMOUS, AG, AP3, and PISTILLATA, pi in Arabidopsis. Class A genes AP2 alone specify sepal development. Class A genes and Class B genes AP3 and PI together specify petals. Class B and Class C AG genes are necessary for stamen formation. Class C genes alone specify carpel formation. When all of these homeotic genes are not expressed in a developing flower, floral parts become leaf-like. The ABC genes code for transcription factors that initiate a cascade of events leading to the actual production of floral parts. In addition to the ABC genes, Class D genes are now being investigated that specifically regulate ovule development. The ovule evolve for the other angiosperm floral parts, and while its development is coordinated with that of the carpel, one would expect more ancient, independent pathways to exist. Senescence. Flower senescence, a developmental program leading to death, are closely linked in many angiosperms. Individual flower petals in some species senesce following pollination. Orchids, which stay fresh for long periods of time if they are not pollinated, are a good example. Fruit ripening and ultimately over-ripening is an example of organ senescence. Whole plant senescence leads to the death of the entire sporophyte generation. Monocarpic plants flower once and then senesce. Polycarpic plants, such as the bristlecone pine, can live thousands of years, 4,900 years is the current record, and flower repeatedly. In polycarpic plants, death is by accident. In monocarpic plants, it appears to be genetically programmed. Flowers and fruits play a key role in the process, and their removal can sometimes delay senescence. Some legumes, senescence can be delayed by removing the developing seed. In other words, the embryo may trigger senescence in the parent plant. During flowering and fruit development, nutrients are reallocated from other parts of the plant to support the development of the next generation. The reproductive structures become a nutrient sink, and this can lead to whole plant senescence. Institute for Advanced Studies, Jodhpur 69. E1. Programmed cell death. Programmed cell death (PCD) is death of a cell in any form, mediated by an intracellular program. Necrosis, which is a form of cell death that results from acute tissue injury and provokes an inflammatory response. PCD is carried out in a regulated process which generally confers advantage during an organism's life cycle. PCD serves fundamental functions during both plant and metazoa multicellular animals tissue development. 1. Types. Apoptosis or type 1 cell death. Autophagy death cytoplasmic, characterized by the formation of large vacuoles which eat away organelles in a specific sequence prior to the nucleus being destroyed. Besides 2 types of PCD, other pathways have been discovered. Cell death, or caspase-independent programmed cell death, or necrosis-like programmed cell death. These alternative routes to death are as efficient as apoptosis and can function as either backup mechanisms or the main type of PCD. Death include anaikes, almost identical to apoptosis except in its induction, cornification, a form of cell death exclusive to the eyes, excitotoxicity and Mullerian degeneration. Plant cells undergo particular processes of PCD which are similar to autophagic cell death. However, some common features of PCD are highly conserved in both plants and metazoa. Apoptosis. Apoptosis is a form of programmed cell death in multicellular organisms. titled death (PCD) and involves a series of biochemical events leading to a characteristic cell morphology and death. In more specific terms, a series of biochemical events that led to a variety of morphological changes, including blebbing, changes to the cell membrane such as loss of membrane asymmetry and attachment, cell shrinkage, nuclear fragmentation, chromatin condensation, and chromosomal DNA fragmentation. Processes of disposal of cellular debris whose results do not damage the organism differentiate apoptosis from necrosis. In contrast, necrosis is a form of traumatic cell death that results from acute cellular injury. Apoptosis, in general, confers advantages during an organism's life cycle. First, the differentiation of fingers and toes in a developing human embryo occurs because cells between the fingers apoptose. The result is that the digits are separate. Between 50 billion and 70 billion cells die each day due to apoptosis in the average human adult. For an average child between the ages of 8 and 14, approximately 20 billion to 30 billion cells die a day. Here, this amounts to the proliferation and subsequent destruction of a mass of cells equal to an individual's body weight. Research on apoptosis has increased substantially since the early 1990s. In addition to its importance as a biological phenomenon, defective apoptotic processes have been implicated in an extensive. Between 50 billion and 70 billion cells die each day due to apoptosis in the average human adult. For an average child between the ages of 8 and 14, approximately 20 billion to 30 billion cells die a day. In a year, this amounts to the proliferation and subsequent destruction of a mass of cells equal to an individual's body weight. Research on apoptosis has increased substantially since the early 1990s. In addition to its importance as a biological phenomenon, defective apoptotic processes have been implicated in an extensive variety of diseases. Excessive apoptosis causes hypertrophy, such as in ischemic damage, whereas an insufficient amount results in uncontrolled cell proliferation, such as cancer. 3. Functions. In cell termination, apoptosis can occur when a cell is damaged beyond repair, infected with a virus, or undergoing stress conditions such as starvation. DNA damage from ionizing radiation or toxic chemicals can also induce apoptosis via the actions of the tumor's uppressing gene p53. Decision for apoptosis can come from a cell itself, from a surrounding tissue, or from a cell that is part of the immune system. In these cases, apoptosis functions to remove the damaged cell, preventing it from sapping further nutrients from the organism, or to prevent the spread of viral infection. Apoptosis also plays a role in preventing cancer. If a cell is unable to undergo apoptosis, due to mutation or biochemical inhibition, it can continue dividing and develop into a tumor. For example, infection by papillomaviruses causes a viral gene to interfere with the cell's p53 protein, an important member of the apoptotic pathway. This interference in the apoptotic capability of the cell plays a critical role in the development of cervical cancer. 3.2 Homeostasis. In the adult organism, the number of cells is kept relatively constant through cell death and division. Some must be replaced when they become diseased or malfunctioning, but proliferation must be compensated by cell death. This balancing process is part of the homeostasis required by living organisms to maintain their internal states within some limits. Some scientists have suggested homeodynamics is a more accurate term. The related term allostasis reflects a balance of a more complex nature by the body. Homeostasis is achieved when the rate of mitosis, cell division, in the tissue is balanced by cell death. If this equilibrium is disturbed, one of two potentially fatal disorders occurs. The cells are dividing faster than they die, effectively developing a tumor. The cells are dividing slower than they die, which results in a disorder of cell loss. The organism must orchestrate a complex series of controls to keep homeostasis tightly controlled, a process that is ongoing for the life of the organism and involves many different types of cell signaling. Impairment of any one of these controls can lead to a disease state. For example, dysregulation of signaling pathway has been implicated in several forms of cancer. The pathway, which conveys an anti-apoptotic signal, has been found to be activated in pancreatic adenocarcinoma tissues. Institute for Advanced Studies, JADPR 70. 3.3. Development. Programmed cell death is an integral part of both plant and animal tissue development. Of an organ or tissue is often preceded by the extensive division and differentiation of a particular cell. The resultant mass is then pruned into the correct form by apoptosis. Unlike necrosis, cellular death caused by injury, apoptosis results in cell shrinkage and fragmentation. This allows cells to be efficiently phagocytosed and their components reused without releasing potentially harmful intracellular substances, such as hydrolytic enzymes, for example, into the surrounding tissue. Recent embryos have suggested how selective cell proliferation, combined with selective apoptosis, sculpts developing tissues in vertebrates. During embryo development, lower plates secrete a gradient of the signaling molecule SHISH, and it is this gradient that directs cells to form patterns in the embryonic neural tube. Cells that receive SHISH in a receptor in their membranes called patched 1 PTC1 survive and proliferate, but, in the absence of SHISH, one of the ends of this same PTC1 receptor, the carboxyl terminal, inside the membrane, is cleaved by CASPASE3, an action that exposes an apoptosis-producing domain. During development, apoptosis is tightly regulated and different tissues use different signals for inducing apoptosis. In birds, bone morphogenic proteins BMP signaling is used to induce apoptosis in the interdigital tissue. In Drosophila flies, steroid hormones regulate cell death. Can also induce apoptosis, such as the sex-specific cell death of hermaphrodite-specific neurons in C. elegans males through low TRA1 transcription factor activity. TRA1 helps prevent cell death. 3.4. Lymphocyte development of B lymphocytes and the development of T lymphocytes in the human body is a complex process that effectively creates a large pool of diverse cells to begin with, then weeds out those potentially damaging to the body. This is the mechanism by which the body removes both the ineffective and the potentially damaging immature cells, and in T cells is initiated by the withdrawal of survival signals. Cytotoxic cells are able to directly induce apoptosis in cells by opening up pores in the target's membrane and releasing chemicals that bypass the normal apoptotic pathway. Pore created by the action of secreted perforin, and the granules contain granzyme B, a serine protease that activates a variety of caspases by cleaving aspartate residues. 4. Process. The process is controlled by a diverse range of cell signals, which may originate either extracellularly extrinsic inducers or intracellularly intrinsic inducers. Cellular signals may include toxins, hormones, growth factors, nitric oxide or cytokines, and therefore must either cross the plasma membrane or transduce to affect a response. These This signals negatively induce apoptosis. In this context, the binding and subsequent initiation of apoptosis by a molecule is termed positive, whereas the active repression of apoptosis by a molecule is termed negative. Cellular apoptotic signaling is a response initiated by a cell in response to stress, and may ultimately result in cell suicide. The binding of glucocorticoids, heat, radiation, nutrient deprivation, viral infection, hypoxia and increased intracellular calcium concentration, e.g. by membrane damage, are all factors that can lead to the release of intracellular apoptotic signals by a damaged cell. Cellular components, such as poly-ADP ribose polymerase, may also help regulate apoptosis. Before the actual process of cell death is carried out by enzymes, apoptotic signals must be connected to the actual death pathway by way of regulatory proteins. This step allows apoptotic signals to either culminate in cell death, or be aborted should the cell no longer need to die. However, two main methods of achieving regulation have been identified: targeting mitochondrial functionality, or directly transducing the signal via adapter proteins to the apoptotic mechanisms. The whole preparation process requires energy and functioning cell machinery. 4.1 Mitochondrial regulation. The mitochondria are essential to multicellular life. Without them, the cell ceases to respire aerobically and quickly dies, a fact exploited by some apoptotic pathways. Apoptotic proteins that Institute for Advanced Studies, Jodhpur 71. Target in different ways. They may cause mitochondrial swelling through the formation of membrane pores, or they may increase the permeability of the mitochondrial membrane and cause apoptotic effectors to leak out. There's also a growing body of evidence that indicates that nitric oxide NO is able to induce apoptosis by helping to dissipate the membrane potential of mitochondria and therefore make it more permeable. Mitochondrial proteins known as SMACs, second mitochondria-derived activator of caspases, are released into the cytosol following an increase in permeability. SMAC binds to inhibitor of apoptosis proteins, IAPs, and deactivates them, preventing the IAPs from arresting the apoptotic process and therefore allowing apoptosis to proceed. It also normally suppresses the activity of a group of cysteine proteases called caspases, which carry out the degradation of the cell. Therefore, the actual degradation enzymes can be seen to be indirectly regulated by mitochondrial permeability. Cytochrome C is released from mitochondria due to formation of a channel, MAC, in the outer mitochondrial membrane, and serves a regulatory function as it precedes morphological change associated with apoptosis. Once released, it binds with APAF1 and ATP, which then bind to pro-CASPASE9 to create a protein complex known as an AP-optosome. The AP-optose leaves the procaspase to its active form of CASPASE9, which in turn activates the effector CASPASE3. Itself subject to regulation by various proteins, such as those encoded by the mammalian BCL2 family of anti-apoptotic genes, the homologs of the CED9 gene found in C. elegans. BCL2 proteins are able to promote or inhibit apoptosis either by direct action on MAC or indirectly through other proteins. It is important to note that the actions of some BCL2 proteins are able to halt apoptosis even if cytochrome C has been released by the mitochondria. Signal transduction. Two important examples of the direct initiation of apoptotic mechanisms in mammals include the TNF-induced tumor necrosis factor model and the phosphosligand-mediated model, both involving receptors of the TNF receptor TNFR family coupled to extrinsic signals. TNF is a cytokine produced mainly by activated macrophages, and is a major extrinsic mediator of apoptosis. Most cells in the human body have two receptors for TNF, TNFR1 and TNFR2. TNFR1 has been shown to initiate the pathway that leads to caspase activation via the intermediate membrane proteins TNF receptor-associated death domain TRADD and phos-associated death domain protein FAD. Binding of this receptor can also indirectly lead to the activation of transcription factors involved in cell survival and inflammatory responses. The link TNF and apoptosis shows why an abnormal production of TNF plays a fundamental role in several human diseases, especially in autoimmune diseases. Institute for Advanced Studies, Jodhpur 72. Receptor, also known as APO1 or CD95, binds the FOS ligand, FOS-L, a transmembrane protein part of the TNF family. The interaction with L results in the formation of the death-inducing signaling complex DISC, which contains the FADD, CASPASE8 and CASPASE10. Types of cells. Type 1 process CASPASE8 directly activates other members of the caspase family, and triggers the execution of apoptosis. In other type 2, the FOS disc starts a feedback loop that spirals into increasing release of pro-apoptotic factors from mitochondria and the amplified activation of CASPASE8. TNFR1 and FOS activation in mammalian cells. A balance between pro-apoptotic BAX, BID, BAC, or BAT and anti-apoptotic BCLXL and BCL2 members of the BCL2 family is established. This balance is the proportion of pro-apoptotic homodimers that form in the outer membrane of the mitochondrion. Pro-apoptotic homodimers are required to make the mitochondrial membrane permeable for the release of caspase activators such as cytochrome C and SMAC. Convergence under normal cell conditions of non-apoptotic cells is incompletely understood, but it has been found that a mitochondrial outer membrane protein, VDAC2, interacts with BAC to keep this potentially lethal apoptotic effector under control. When the death signal is received, products of the activation cascade displace VDAC2 and BAC is able to be activated. This is the caspase-independent apoptotic pathway that is mediated by AIF, apoptosis-inducing factor. Pathways and signals lead to apoptosis. There is only one mechanism that actually causes the death of a cell in this process. After the appropriate stimulus has been received by the cell and the necessary controls exerted, a cell will undergo the organized degradation of cellular organelles by activated proteolytic caspases. A cell undergoing apoptosis shows a characteristic morphology that can be observed with a microscope. Cell shrinkage and rounding due to the breakdown of the proteinaceous cytoskeleton by caspases. The cytoplasm appears dense, and the organelles appear tightly packed. Chromatin undergoes condensation into compact patches against the nuclear envelope in a process known as pycnosis, a hallmark of apoptosis. The nuclear envelope becomes discontinuous and the DNA inside it is fragmented in a process referred to as karyorrhexis. The nucleus breaks into several discrete chromatin bodies or nucleosomal units due to the degradation of DNA. The cell membrane shows irregular buds known as blebs. The cell breaks up into several vesicles called apoptotic bodies, which are then phagocytosed. Apoptosis progresses quickly and its products are quickly removed, making it difficult to detect or visualize. During karyorrhexis, endonuclease activation leaves short DNA fragments, regularly spaced in size. These give a characteristic, laddered, appearance on agar gel after electrophoresis. Tests for DNA laddering differentiate apoptosis from ischemic or toxic cell death. Or removal of dead cells. Dying cells that undergo the final stages of apoptosis display phagocytotic molecules, such as phosphatidylserine, on their cell surface. Is normally found on the cytosolic surface of the plasma membrane, but is redistributed during apoptosis to the extracellular surface by a hypothetical protein known as scramblase. These molecules mark the cell for phagocytosis by cells possessing the appropriate receptors, such as macrophages. Upon recognition, the phagocyte reorganizes its cytoskeleton for engulfment of the cell. The removal of dying cells by phagocytes occurs in an orderly manner without eliciting an inflammatory response. 5. Implication in disease. 5. Apoptotic pathways. The many different types of apoptotic pathways contain a multitude of different biochemical components, many of them not yet understood. A pathway is more or less sequential in nature. It is a victim of causality. Removing or modifying one component leads to an effect in another. In a living organism this can have disastrous effects, often in the form of disease or disorder. A discussion of every. Institute for Advanced Studies, Jodhpur 73. By modification of the various apoptotic pathways would be impractical, but the concept overlying each one is the same. The normal functioning of the pathway has been disrupted in such a way as to impair the ability of the cell to undergo normal apoptosis. Results in a cell that lives past its use-by date and is able to replicate and pass on any faulty machinery to its progeny, increasing the likelihood of the cell becoming cancerous or disease. A recently described example of this concept in action can be seen in the development of a lung cancer called NCI-H460. The X-linked inhibitor of apoptosis protein, XIAP, is overexpressed in cells of the H460 cell line. XIAPs bind to the processed form of CASPASE9 and suppress the activity of apoptotic activator cytochrome C, therefore overexpression leads to a decrease in the amount of pro-apoptotic agonists. The consequence, the balance of anti-apoptotic and pro-apoptotic effectors is upset in favor of the former, and the damaged cells continue to replicate despite being directed to die. 5-2p53 dysregulation. The tumor suppressor protein p53 accumulates when DNA is damaged due to a chain of biochemical reactions. Alpha interferon and beta interferon, which induce transcription of the p53 gene and result in the increase of p53 protein level and enhancement of cancer cell apoptosis. P prevents the cell from replicating by stopping the cell cycle at G1, or interphase, to give the cell time to repair. However, it will induce apoptosis if damage is extensive and repair efforts fail. Any disruption to the regulation of the p53 or interferon genes will result in impaired apoptosis and the possible formation of tumors. 5.3 HIV progression. Progression of the human immunodeficiency virus, HIV, to AIDS is primarily due to the depletion of CD4 plus T helper lymphocytes, which leads to a compromised immune system. Is by which T helper cells are depleted is apoptosis, which can be the end product of multiple biochemical pathways. Enzymes inactivate anti-apoptotic BCL2 and simultaneously activate pro-apoptotic PROCASPASE8. This does not directly cause cell death but primes the cell for apoptosis should the appropriate signal be received. HIV products may increase levels of cellular proteins which have a promotive effect on Fas-mediated apoptosis. HIV proteins decrease the amount of CD4 glycoprotein marker present on the cell membrane. Released viral particles and proteins present in extracellular fluid are able to induce apoptosis in nearby, bystander, T helper cells. HIV production of molecules involved in marking the cell for apoptosis, giving the virus time to replicate and continue releasing apoptotic agents and virions into the surrounding tissue. The NP4+ cell may also receive the death signal from a cytotoxic T cell, leading to apoptosis. In addition to apoptosis, infected cells may also die as a direct consequence of the viral infection. 5.4 Infection. Viruses can trigger apoptosis of infected cells via a range of mechanisms including receptor binding, activation of protein kinase R, PKR, interaction with P53, expression of viral proteins coupled to MHC proteins on the surface of the infected cell, allowing recognition by cells of the immune system, such as natural killer and cytotoxic T cells, that then induce the infected cell to undergo apoptosis. Most viruses encode proteins that can inhibit apoptosis. Several viruses encode viral homologs of DL2. These homologs can inhibit pro-apoptotic proteins such as BAX and BAC, which are essential for the activation of apoptosis. Examples of viral BCL2 proteins include the Epstein-Barr virus BHRF1 protein and the adenovirus E1b19K protein. Some viruses express caspase inhibitors that inhibit caspase activity, and an example is the Korma protein of cowpox viruses. A number of viruses can block the effects of TNF and FOS. For example, the MT2 protein of myxoma viruses can bind TNF, preventing it from binding the TNF receptor and inducing a response. Furthermore, many viruses express p53 inhibitors that can bind p53 and inhibit its transcriptional transactivation activity. Consequently, p53 cannot induce apoptosis since it cannot induce the expression of pro-apoptotic proteins. The virus E1b55K protein and the hepatitis B virus HVX protein are examples of viral proteins that can perform such a function. Interestingly, viruses can remain intact from apoptosis, particularly in the latter stages of infection. Transported in the apoptotic bodies that pinch off from the surface of the dying cell, and the fact that they are engulfed by phagocytes prevents the initiation of a host response. This favors the spread of the virus. Programmed cell death in plant tissue. It regulates vascular tissue identity in Arabidopsis. Bonk and colleagues state that one of the two long-distance transport systems in vascular plants, xylem, consists of several cell types, the differentiation of which involves deposition of elaborate cell wall thickenings and programmed cell death. The authors emphasize that the products of plant PCD play an important structural role. Basic morphological and biochemical features of PCD have been conserved in both plant and animal kingdoms. It should be noted, however, that specific types of plant cells carry out unique cell death programs. These are apoptosis, for instance, nuclear DNA degradation, but they also have their own peculiarities, such as nuclear degradation being triggered by the collapse of the vacuole and trichyre elements of the xylem. Janet Bach and Christopher J. Lever, of the Department of Plant Sciences, University of Oxford, carried out research on mutations in the mitochondrial genome of sunflower cells. Results suggest that mitochondria play the same key role in vascular plant PCD as in other eukaryotic cells. Institute for Advanced Studies, Jodhpur 74. CD in pollen prevents inbreeding. Plants enforce self-incompatibility, SI, is an important means to prevent self-fertilization. Research on corn papaverrhias has revealed that proteins in the pistil on which the pollen lands interact with pollen and trigger PCD and incompatible, i.e. self-pollen. Researchers Stephen G. Thomas and Veronica E. Franklin Tong also found that the response involves rapid inhibition of pollen tube growth, followed by PCD. Ground cell death in slime molds. The slime mold dictates the peculiarity of adopting either a predatory amoeba-like behavior in its unicellular form, or coalescing into a mobile slug-like form when dispersing the spores which will give birth to the next generation. The of dead cells which have undergone a type of PCD that shares many features of an autophagic cell death, massive vacuoles forming inside cells, a degree of chromatin condensation, but no denafragmentation. The structural role of the residues left by the dead cells is reminiscent of the products of PCD in plant tissue. D. difficium is a slime mold, part of a branch which may have emerged from eukaryotic ancestors about 1 billion years before the present. They apparently emerged after the ancestors of green plants and the ancestors of fungi and animals had differentiated. Place of entry. The fact that PCD has been observed in the humble, simple, 6-chromosome D discoideum has additional significance. It permits the study of a developmental PCD path which does not depend on the caspases which are characteristic of apoptosis. 9. Evolutionary origin of PCD. Biologists have long suspected that mitochondria originated from bacteria which had been incorporated as endosymbionts living together inside of larger eukaryotic cells. It was Lynn Margulis who from 1967 on championed this theory, which has since become widely accepted. The most convincing evidence for this theory is the fact that mitochondria possess their own DNA and are equipped with genes and replication apparatus. This evolution step would have been more than risky for the primitive eukaryotic cells which began to engulf the energy-producing bacteria and conversely, a perilous step for the ancestors of mitochondria which began to invade their proto-eukaryotic hosts. This process is still evident today, between human white blood cells and bacteria. Invading bacteria are destroyed by the white blood cells, however, it is not uncommon for the chemical warfare waged by prokaryotes to succeed, with the consequence known as infection by its resulting damage. Evolutionary events, about 2 billion years before the present, made it possible for certain eukaryotes and energy-producing prokaryotes not only to coexist, but to mutually benefit from their symbiosis. Mitochondriate eukaryotic cells live poised between life and death, because mitochondria still retain the repertoire of molecules which can trigger cell suicide. This process has now been evolved to happen only when programmed. In signals to cells, such as feedback from neighbors, stress or DNA damage, mitochondria release caspase activators which trigger the cell death-inducing biochemical cascade. As such, the cell suicide mechanism is now crucial to all of our lives. EU. Senescence and aging. Senescence refers to the biological processes of a living organism approaching an advanced age, i.e., the combination of processes of deterioration which follow the period of development of an organism. Senescence is derived from the Latin word senex, meaning old man, or old age, or advanced in age. 1. Cellular senescence. Cellular senescence is a phenomenon where normal diploid differentiated cells lose the ability to divide, normally after about 50 cell divisions in vitro. Some cells become senescent before because of DNA double-strand breaks, toxins, etc. This is also known as replicative senescence, the Hayflick phenomenon, or the Hayflick limit in honor of Dr. Leonard Hayflick, who was the first to publish this information in 1965. To DNA damage, including shortened telomeres, cells either age or self-destruct, apoptosis. Programmed cell death if the damage cannot be repaired. In this cellular suicide, the death of one, or more, cells may benefit the organism as a whole. In plants, the death of the water-conducting xylem cells, tracheids and vessel elements, allows the cells to function more efficiently and so deliver water to the upper parts of a plant. 2. Aging of the whole organism. Organismal senescence is the aging of whole organisms. The term aging has become so commonly equated with senescence that the terms will be used interchangeably in this article. Aging is generally characterized by the declining ability to respond to stress, increasing homeostatic imbalance and increased risk of aging-associated diseases. Because of this, death is the ultimate consequence of aging. Differences in maximum lifespan among species correspond to different rates of aging. For example, inherited differences in the rate of aging make a mouse elderly at 3 years and a human elderly at 90 years. Genetic differences affect a variety of physiological processes, including the efficiency of DNA repair, antioxidant enzymes, and rates of free radical production. Institute for Advanced Studies, Jodhpur 75. Senescence of the organism gives rise to the Gompertz-Makeham law of mortality, which says that mortality rate rises rapidly with age. Some animals, such as some reptiles and fish, age slowly. Some even exhibit negative senescence, in which mortality falls with age, in disagreement with the Gompertz-Makeham law. Whether cell senescence, Hayflick limit, plays a causative role in organismal aging is at present and is an ongoing area of active investigation. 3. Theories of aging. The process of senescence is complex, and may derive from a variety of different mechanisms and exist for a variety of different reasons. However, senescence is not universal, and scientific evidence suggests that cellular senescence evolved in certain species as a mechanism to prevent the onset of cancer. In a few simple species, senescence is negligible and cannot be detected. All such species are postmeiotic cells. They reduce the effect of damaging free radicals by cell division and dilution. Such species are not immortal, however, as they will eventually fall prey to trauma or disease. Moreover, average lifespans can vary greatly within and between species. This suggests that both genetic and environmental factors contribute to aging. Traditionally, theories that explain senescence have generally been divided between the programmed and stochastic theories of aging. Programmed theories imply that aging is regulated by biological clocks operating throughout the lifespan. This regulation on changes in gene expression that affect the systems responsible for maintenance, repair and defense responses. Stochastic mental impacts on living organisms that induce cumulative damage at various levels as the cause of aging, examples which range from damage to DNA, damage to tissues and cells by oxygen radicals widely known as free radicals countered by the even more well-known antioxidants, and cross-linking. Conversion is a progressive failure of homeodynamics, homeostasis, involving genes for the maintenance and repair, stochastic events leading to molecular damage and molecular heterogeneity, and chance events determining the probability of death. Interacting systems of maintenance and repair comprise the homeodynamic, old term, homeostasis space of a biological system. Aging is considered to be a progressive shrinkage of homeodynamic space mainly due to increased molecular heterogeneity. 3.1 Theories. Aging is believed to have evolved because of the increasingly smaller probability of an organism still being alive at older age, due to predation and accidents, both of which may be random and age invariant. These strategies which result in a higher reproductive rate at a young age, but shorter overall lifespan, result in a higher lifetime reproductive success and are therefore favored by natural selection. Essentially, aging is therefore the result of investing resources in reproduction, rather than maintenance of the body. That disposable soma theory, in light of the fact that accidents, predation and disease will eventually kill the organism no matter how much energy is devoted to repair of the body. Various other, or more specific, theories of aging exist, and are not necessarily mutually exclusive. The geneticist J.B.S. Haldane wondered why the dominant mutation which causes Huntington's disease remained in the population, why natural selection had not eliminated it. The onset of this neurological disease is, on average, at age 45 and is invariably fatal within 10 to 20 years. Haldane assumed, probably reasonably, that in human prehistory, few survived until age 45. Since his contribution to the next generation was therefore small relative to the large cohorts of younger age groups, the force of selection against the clade-acting deleterious mutations was correspondingly small. However, if a mutation affected younger individuals, selection against it would be strong. Therefore, late-acting deleterious mutations could accumulate in populations over evolutionary time through genetic drift. This principle has been demonstrated experimentally. These later-acting deleterious mutations which are believed to cause, or perhaps more correctly allow, age-related mortality. Peter Medawar formalized this observation in his mutation accumulation theory of aging. The force of selection weakens with increasing age, even in a theoretically immortal population, provided only that it is exposed to real hazards of mortality. If a genetic disaster happens late enough in individual life, its consequences may be completely unimportant. The real hazards of mortality are typically predation, disease, accidents. So, even in a mortal population, whose fertility does not decline with time, will have fewer individuals alive in older age groups. This is called extrinsic mortality. Not depleted in numbers yet by extrinsic mortality, contribute far more to the next generation than the few remaining older cohorts, so the force of selection against late-acting deleterious mutations, which only affect these few older individuals, is very weak. Mutations may not be selected against, therefore, and may spread over evolutionary time into the population. The main testable prediction made by this model is that species which have high extrinsic mortality in nature will age more quickly and have shorter intrinsic lifespans. This is borne out among mammals, the most well-studied in terms of life history. There is a correlation almost between body size and lifespan, such that larger species live longer than smaller species in controlled, optimum conditions, but there are notable exceptions. For instance, many bats and rodents are similarly sized, yet bats live much, much longer. For instance, the little brown bat, half the size of a mouse, can live 30 years in the wild. A mouse will live 2 to 3 years even with optimum conditions. The explanation is that bats have fewer predators, so therefore low extrinsic mortality. Thus more will survive to later ages, so the force of selection against late-acting deleterious mutations is stronger. Fewer late-acting deleterious mutations equals slower aging equals longer lifespan. Birds also warm-blooded and similarly sized to many small mammals, yet live often 5 to 10 times long. They clearly. Institute for Advanced Studies, Jodhpur 76. Have fewer predation pressures compared with ground-dwelling mammals. And seabirds, which generally have the fewest predators of all birds, live longest. Also, in the body size versus lifespan relationship, predator mammals tend to have longer lifespans than prey animals in a controlled environment such as a zoo or nature reserve. The explanation for the long lifespans of primates such as humans, monkeys and apes relative to body size is that their intelligence and often sociality helps them avoid becoming prey. Being a predator, being smart and working together all reduce extrinsic mortality. Another evolutionary theory of aging was proposed by George C. Williams and involves antagonistic pleiotropy. A single gene may affect multiple traits. Traits that increase fitness early in life may also have negative effects later in life. But individuals are alive at young ages and at old ages, even small positive effects early can be strongly selected for, and large negative effects later may be very weakly selected against. Just following example, perhaps a gene codes for calcium deposition in bones which promotes juvenile survival and will therefore be favored by natural selection. However, this same gene promotes calcium deposition in the arteries, causing negative effects in old age. Therefore, negative effects in old age may reflect the result of natural selection for pleiotropic genes which are beneficial early in life. In this case, fitness is relatively high when Fisher's reproductive value is high and relatively low when Fisher's reproductive value is low. 3.2 A number of genetic components of aging have been identified using model organisms, ranging from the simple budding yeast Saccharomyces cerevisiae to worms such as Caenorhabditis elegans and fruit flies Drosophila melanogaster. Study of these organisms has revealed the presence of at least 2 conserved aging pathways. One of these pathways involves the gene SIR2, a NAV+-dependent histone deacetylase. SIR2 is required for genomic silencing at 3 loci, the yeast mating loci, the telomeres in the ribosomal DNA, rDNA. In some cases, shifting may be partially caused by homologous recombination between rDNA repeats. Excision of rDNA repeats results in the formation of extrachromosomal rDNA circles, ERCs. ERCs replicate and preferentially segregate to the mother cell during cell division, and are believed to result in cellular senescence by titrating away competing for essential nuclear factors. In other species, nor even all strains of the same yeast species of yeast which also display replicative senescence, and ERCs are not believed to contribute to aging in higher organisms such as humans. They have not been shown to accumulate in mammals in a similar manner to yeast. Extrachromosomal circular DNA (ECCDNA) has been found in worms, flies, and humans. The origin and role of ECCDNA in aging, if any, is unknown. Despite DNA in aging in higher organisms, extra copies of SIR2 are capable of extending the lifespan of both worms and flies, though in flies, this finding has not been replicated by other investigators, and the activator of SIR2, resveratrol, does not reproducibly increase lifespan in either species. Whether the SIR2 bugs in higher organisms have any role in lifespan is unclear, but the human SIRT1 protein has been demonstrated to desilate P53, Cu70, and the FRCAD family of transcription factors. SIRT1 can also regulate acetylates such as CBP, P300, and has been shown to desilate specific histone residues. RAS1 and RAS2 also affect aging in yeast and have a human homologue. RAS2 overexpression has been shown to extend lifespan in yeast. Other genes regulate aging in yeast by increasing the resistance to oxidative stress. Superoxide dismutase, a protein that protects against the effects of mitochondrial free radicals, can extend yeast lifespan in stationary phase when overexpressed. In higher organisms, aging is likely to be regulated in part through the insulin IGF-1 pathway. Mutations that affect insulin-like signaling in worms, flies, and the growth hormone IGF-1 axis in mice are associated with extended lifespan. In yeast, SIR2 activity is regulated by the nicotinamidase PNC1. PNC1 is transcriptionally upregulated under stressful conditions such as caloric restriction, heat shock, and osmotic shock. By converting nicotinamide to niacin, it removes nicotinamide, which inhibits the activity of SIR2. A nicotinamide found in humans, known as PBEF, may serve a similar function, and a secreted form of PBEF known as vispadin may help to regulate serum insulin levels. It is not known, however, whether these mechanisms also exist in humans since there are obvious differences in biology between humans and model organisms. SIR2 activity has been shown to increase under calorie restriction. Due to the lack of available glucose in the cells, more NAD+ is available and can activate SIR2. Resveratrol, a polyphenol found in the skin of red grapes, was reported to extend the lifespan of yeast, worms, and flies. The lifespan extension in flies and worms have proved irreproducible by independent investigators. It has been shown to activate SIR2 and therefore mimics the effects of calorie restriction, if one accepts that caloric restriction is indeed dependent on SIR2. Expression is imperfectly controlled, and it is possible that random fluctuations in the expression levels of many genes contribute to the aging process as suggested by a study of such genes in yeast. Insects, which are genetically identical, nonetheless can have substantially different responses to outside stimuli, and markedly different lifespans, indicating the epigenetic factors play an important role in gene expression and aging as well as genetic factors. 3.3. Cellular senescence. As noted above, senescence is not universal, and senescence is not observed in single-celled organisms that reproduce through the process of cellular mitosis. Moreover, cellular senescence is not observed in many organisms, including perennial plants, sponges, corals, and lobsters. Those species where cellular senescence is observed, cells eventually become postmeiotic when they can no longer replicate themselves through the process of cellular mitosis, i.e. Institute for Advanced Studies, Jodhpur 77. Cells experience replicative senescence. How and why cells become postmeiotic in some species has been the subject of much research and speculation, but as noted above, it is widely believed that cellular senescence evolved as a way to prevent the onset and spread of cancer. Somatic cells that have divided many times will have accumulated DNA mutations and would therefore be in danger of becoming cancerous if cell division continued. Lately, the role of telomeres in cellular senescence has aroused general interest, especially with a view to the possible genetically adverse effects of cloning. The successive shortening of the chromosomal telomeres with each cell cycle is also believed to limit the number of divisions of the cell, thus contributing to aging. There have, on the other hand, also been reports that cloning could alter the shortening of telomeres. Some cells do not age and are therefore described as being biologically immortal. It is by some that when it is discovered exactly what allows these cells, whether it be the result of telomere lengthening or not, to divide without limit, that it will be possible to genetically alter other cells to have the same capability. It is further theorized that it will eventually be possible to genetically engineer all cells in the human body to have this capability by employing gene therapy and thereby stop or reverse aging, effectively making the entire organism potentially immortal. Cancer cells are usually immortal. This evasion of cellular senescence is the result, in about 85% of tumors, of upactivation of their telomerase genes. This simple observation suggests that reactivation of telomerases in healthy individuals could greatly increase their cancer risk. Whether cell senescence plays any role in organismal aging is at present unknown, and is an active area of investigation. Mice lacking telomerase do not immediately show accelerated aging. 3.4 Catch. Earliest aging theory was the rate of living hypothesis described by Raymond Pearl in 1928, based on the idea that fast basal metabolic rate corresponds to short maximum lifespan, much as a rapidly running machine will experience more damage from wear. The idea had been posited earlier by Max Rubner. This theory, in the form of various types of specific damage detailed below which, all other things being equal, may reduce lifespan. In general, this theory does not adequately explain the differences in lifespan either within, or between, species. Fully restricted animals process as much, or more, calories per gram of body mass, as their ad libitum-fed counterparts, yet exhibit substantially longer lifespans. Metabolic rate is a poor predictor of lifespan for birds. Bats and other species which presumably have reduced mortality from predation, and therefore have evolved long lifespans even in the presence of very high metabolic rates. Specific types of chemical damage caused by metabolism, it is suggested that damage to long-lived biopolymers, such as structural proteins or DNA, caused by ubiquitous chemical agents in the body such as oxygen and sugars, are in part responsible for aging. The damage can include breakage of biopolymer chains, cross-linking of biopolymers, or chemical attachment of unnatural substituents, haptens, to biopolymers. Under normal hormones, approximately 4% of the oxygen metabolized by mitochondria is converted to superoxide ion, which can subsequently be converted to hydrogen peroxide, hydroxyl radical, and eventually other reactive species including other peroxides and singlet oxygen, which can in turn generate free radicals capable of damaging structural proteins and DNA. Certain metal ions found in the body, such as copper and iron, may participate in the process. In Wilson's disease, a hereditary defect which causes the body to retain copper, some of the symptoms resemble accelerated senescence. These processes are termed oxidative damage and are linked to the benefits of nutritionally derived polyphenol antioxidants. Sugars such as glucose and fructose can react with certain amino acids such as lysine and arginine and certain DNA bases such as guanine to produce sugar adducts, in a process called glycation. These can further rearrange to form reactive species which can then cross-link the structural proteins or DNA to similar biopolymers or other biomolecules such as non-structural proteins. People with diabetes, who have elevated blood sugar, develop senescence-associated disorders much earlier than the general population, but can delay such disorders by rigorous control of their blood sugar levels. There is evidence that sugar damage is linked to oxidant damage in a process termed glycoxidation. Free radicals can damage proteins, lipids or DNA. Glycation mainly damages proteins. Damaged proteins and lipids accumulate in lysosomes as lipofuscin. Damage to structural proteins can lead to loss of function. For example, damage to collagen of blood vessel walls can lead to vessel wall stiffness and thus hypertension, and vessel wall thickening and reactive tissue formation, atherosclerosis. Similar processes in the kidney can lead to renal failure. Damage to enzymes reduces cellular functionality. Lipid peroxidation of the inner mitochondrial membrane reduces the electric potential and the ability to generate energy. It is probably no accident that nearly all of the so-called accelerated aging diseases are due to defective DNA repair enzymes. It is believed the effect of alcohol on aging can be partly explained by alcohol's activation of the HPA axis, which stimulates glucocorticoid secretion, long-term exposure to which produces symptoms of aging. 3.5 Reliability theory. Reliability theory suggests that biological systems start their adult life with a high load of initial damage. Reliability theory is a general theory about systems failure. It allows researchers to predict the age-related failure kinetics for a system of given architecture, reliability structure, and given reliability of its components. Reliability predicts that even those systems that are entirely composed of non-aging elements with a constant failure rate will nevertheless deteriorate, fail more often with age, if these systems. Institute for Advanced Studies, Jodhpur 78. Or redundant and irreplaceable elements. Aging, therefore, is a direct consequence of systems redundancy. Reliability theory also predicts the late life mortality deceleration with subsequent leveling off, as well as the late life mortality plateaus, as an inevitable consequence of redundancy exhaustion at extreme old ages. Theory explains why mortality rates increase exponentially with age, the Gompertz law, in many species, by taking into account the initial flaws defects in newly formed systems. It also explains why organisms prefer to die according to the Gompertz law, while technical devices usually fail according to the Weibull-Pauwart law. Reliability theory allows to specify conditions when organisms die according to the Weibull distribution. Organisms should be relatively free of initial flaws and defects. The theory makes it possible to find a general failure law applicable to all adult and extreme old ages, where the Gompertz and the Weibull laws are just special cases of this more general failure law. The theory of relative differences in mortality rates of compared populations within a given species vanish with age. Compensation law of mortality and mortality convergence is observed due to the exhaustion of initial differences in redundancy levels. 3.6 Neuroendocrine immunological theories. Senescence may also simply be a result of wear and tear overwhelming repair mechanisms. Cells posit senescence is a mechanism to control the development and spread of cancer. If cells have built-in limits to how many times they can replicate, they must somehow overcome this before they can spread indefinitely. Recently, early senescence has been alleged to be a possible unintended outcome of early cloning experiments. Most notably, the issue was raised in the case of Dolly the sheep, following her death from a contagious lung disease. The claim that involved premature senescence has been vigorously contested, e.g. by Carrie Lynn McIntosh in her book, Illegal Beings, Human Clones in the Law, and Dolly's creator, Dr. Ian Wilmot, has expressed the view that her illness and death were probably unrelated to the fact that she was a clone. A set of rare hereditary genetic disorders, each called progeria, has been known for some time. Sufferers exhibit symptoms resembling accelerated aging, including wrinkled skin. The cause of Hutchinson-Gilford progeria syndrome was reported in the journal Nature in May 2003. This report suggests that DNA damage, not oxidative stress, is the cause of this form of accelerated aging. Unit 6. Institute for Advanced Studies, Jodhpur 1. A. Photosynthesis, light harvesting complexes, mechanisms of electron transport, photoprotective mechanisms, CO. 2. Fixation C. 3. C. 4. And CAM pathways. Photosynthesis is essentially the only mechanism of energy input in the living world. Photosynthesis light, synthesis putting together, is an anabolic process of manufacture of organic compounds inside the chlorophyll-containing cells from carbon dioxide and water with the help of sunlight as a source of energy. A simple equation of photosynthesis is as follows. Chlorophyll 6CO2 plus 16H2OC6H12O6 plus 6O2 radiant energy. However, the function of water is to provide hydrogen for the synthesis of organic compounds. All the liberated oxygen comes from it. Therefore, the equation is modified as chlorophyll 6CO2 plus 12H2OC6H12O6 plus 6H2O plus 6O2 radiant energy. The source of molecular oxygen was water and not carbon dioxide, as was believed earlier was experimentally proved first by Robert Hill, 1937, and later confirmed by M. D. Kamen and S. Rubin, 1945, employing tracer technique in which heavy isotopes of oxygen 18O were used. But this experimental proof was based on the suggestions of C. B. Van Niel's work on bacterial photosynthesis, 1930. Suggested that in green plants H2O is the source of reduction, and when split yields H and O and O2 released by plants is derived from water, not from CO2. This splitting of water and light by green plants has come to be known as photolysis of water and the theory, Van Niel's theory of photolysis of water. Chloroplasts, structures and photosynthetic pigments. Chloroplast is the seat of photosynthesis and is best exemplified in the higher plants. A chloroplast is covered by an envelope of two membranes which are separated by paraplastidial space of 10 to 20 nm. Internally, a chloroplast contains a matrix or stroma in which are embedded a number of flattened membranous sacs called thylakoids or lamella. The external surface of thylakoids contains the photosynthetic pigments and serves the ends of light reaction. The stroma, on the other hand, is concerned with the events of the dark reaction. In certain regions the thylakoids are stacked to form grana. The longer thylakoids that connect one granum to another extend through the stroma, so these membranes are usually referred to as stroma thylakoids. Photosynthetic pigments. The photosynthetic pigments present in thylakoid membranes consist largely of two kinds of green chlorophylls, chlorophyll 8 C55H7005 N4 megagrams, and chlorophyll beet C55H7206 N4 megagrams. Also present are yellow to orange pigments classified as carotenoids. There are two kinds of carotenoids, the pure hydrocarbon keratines and the oxygen-containing xanthophylls. Certain carotenoids, especially violaxanthin, the xanthophyll, also exist in the chloroplast envelope, giving it a yellowish color. In most plants, including green algae, beta-carotene and lutein are the most abundant carotenoids in the thylakoids. Events of photosynthesis. Photosynthesis consists of two types of reactions, a light-dependent one and a light-independent one. The light-dependent reaction is a photochemical reaction or light reaction as it came to be called, culminating in the generation of NADPH2, ATP and evolution of molecular oxygen. The NADPH2 and ATP are energy-rich, having caught the electrons that became available when light impinged upon chlorophyll. They form the assimilatory power, utilized for County 2 fixation. The events of CO2 fixation is light-independent reaction and is designated as dark reaction. Light reaction. Light reaction consists of two phases. Phase 1 energy absorption and retention of light by the photosynthetic pigments and Phase 2 energy transduction, conversion of light energy absorbed in Phase 1 into chemical energy ATP and NADPH2 by photophosphorylation. Phase 1 energy absorption. Photosynthetic units. The events of light reaction are mediated through photosynthetic units, a photosynthetic unit being the smallest group of pigment molecules, together with their lipoprotein associate substances, able to bring about a photochemical act, photoact. The term Institute for Advanced Studies, Jodhpur 2. Photochemical act means absorption and migration of a light quantum by a trapping center, as a result of which an electron is released. Emmer thought that a photosynthetic unit PSU contained at least 2,500 chlorophyll molecules, but recent work by Bessel-Koch indicates that a photosynthetic unit contains only about 250 chlorophyll molecules. The occurrence of PSU as a distinct morphological entity was obtained by Park and his co-workers and they christened it quantosome. Absorption of light by pigments. All the light incident on photosynthesizing surface is not used for photosynthesis. Much of it is lost and quite some is reflected back. Again, only a small fraction of the absorbed light is used to drive photosynthesis. It is estimated that in full sunlight, just about 3% is used for photosynthetic purpose. Depending upon the pigment composition, the various plant groups absorb and utilize light of different spectral regions. Most green plants absorb light in the visible spectrum, 390-700 nanometers, whereas purple bacteria employ wavelengths ranging from near ultraviolet to infrared, 800-950 nanometers. This range of the spectrum through which photosynthesis can take place is called photosynthetically active radiation, but the entire range is not employable in photosynthesis. The green plants, for example, absorb light maximally in the red and blue regions of the spectrum. Study of the absorption spectra shows the quantitative relationship between the wavelength of light and its absorption by the pigment in question. Thus, Weissfeld has its absorption peaks at 660 nanometers and 430 nanometers, chlorophyll B at 648 nanometers and 456 nanometers, carotene at 478 nanometers and 449 nanometers, and xanthophyll same as carotene. Light trap. Chlorophyll utilizes the light that it absorbs on its own and also the light transferred to it by other pigments. This funneling of light from other pigments to chlorophyll has been called light trap or light sink. The light trap makes for a much better light harvesting efficiency, for it ensures funneling of light quantity towards one acceptor molecule of chlorophyll. The light reaction actually consists of two photochemical reactions which are separated both in time and space. They are designated as photoact I and photoact II, and the two reactions are mediated by two different systems whose composition differ in terms of pigments, electron carriers and light trap mechanisms. The mediating agencies of the two photoacts are respectively called photosystem I and photosystem II. Photosystems. The concept of two photosystems originated in the work of Emerson and Lewis, 1943. Working spectrum for the pigments of chlorella, they found that at wavelengths of light between 600 and 680 nanometers, wavelengths corresponding to the red region of the spectrum, the evolution of oxygen was at its maximum. Light of wavelengths beyond 680 nanometers, a region of the spectrum referred to as far red, was supplied. There was a drop in the evolution of oxygen, indicative of lessened photosynthesis efficiency. This observation had been christened the red drop. His group found that if light of shorter wavelengths was provided at the same time as the longer red wavelengths, photosynthesis was even. Institute for Advanced Studies, Jodhpur 3. Faster than when either of wavelengths alone was provided, the synergism or enhancement became known as the Emerson enhancement effect. These two, the red drop effect and enhancement effect, led to the first indication that the light reaction has two sites of action, one in the red region of the spectrum and the other in the far. The explanation for these two effects is that the light reaction actually consists of two photoacts, photoact I and photoact II, mediated by two photosystems, photosystem I and photosystem II. Photosystem I is driven by the far red light, and when it operates alone, it produces the red drop effect. But when it operates along with photosystem II, which functions in the red region, enhancement effect is produced. Physical separation of the two photosystems had been successfully carried out and their functions clarified. Photosystem I has been located in the thylakoid membranes. It is made of forms of chlorophyll A, one absorbing maximally at 683 nanometers, the second absorbing maximally at 695 nanometers, and the third at 670 nanometers. The last of these has been called P700. Photosystem II had been located in the stroma thylakoids. It is made up of two forms of chlorophyll with maximum absorption at 670 and 690 nanometers. The second form is christened P690. Each photosystem components. A reaction center made up of a special chlorophyll molecule. In photosystem I it is a protein-bound chlorophyll molecule, P700. In photosystem II it is P690. The reaction centers are the actual sites where light energy is converted to chemical energy. Electron carriers in photosystem I, X, plastocyanin, cytochrome F and ferredoxin is the electron carrier. Photosystem II has plastoquinone and cytochrome B559. Other chlorophyll and carotenoids, which merely serve to transfer the light absorbed by them to the active centers. Photosystem I takes part in both cyclic and non-cyclic photophosphorylations. PSI can carry on cyclic photophosphorylation independently. Normally it drives an electron from photosystem II to NADP+. Photosystem II picks up electron release during photolysis of water. The same is extruded on absorption of light energy. As the activated electron passes over cytochrome complex, sufficient energy is released to take part in the synthesis of ATP from ADP and inorganic phosphate. This photophosphorylation is not. PS2 can operate only in conjunction with PSI. Phase 2, energy transduction. The excited molecules of P700 and P690 transduce their energies to generate ATP and NADPH2. Molecular oxygen is also produced but it escapes out of the photosynthetic system. ATP and NADPH2 together constitute the assimilatory power and are employed in the fixation of CO2 in the dark reaction. Photophosphorylation. Photophosphorylation is the light-driven or light-energized synthesis of ATP. It was discovered by Amen et al. In 1954. Photophosphorylation is of two main types, cyclic and non-cyclic. Cyclophosphorylation. It is a process of photophosphorylation in which an electron expelled by the excited photocenter is returned to it after passing through a series of electron carriers. Cyclic photophosphorylation is performed by photosystem I only. Its photocenter P700 extrudes an electron with a gain of 23 kilocalories per mole of energy after absorbing a photon of light Hb. After losing the electron, the photocenter becomes oxidized. The electron passes through a series of carriers including X, ferredoxin, plastoquinone, cytochrome complex and plastocyanin before returning to photocenter. While between ferredoxin and plastoquinone and, or over the cytochrome complex, the electron loses sufficient energy to form ATP from ADP and inorganic phosphate. Institute for Advanced Studies, JADPR 4. Halobacteria or halophile bacteria also perform photophosphorylation, but ATP thus produced is not used in synthesis of food. These bacteria possess purple pigment bacteria or rhodopsin attached to plasma membrane. As it falls on the pigment, it creates a proton pump which is used in ATP synthesis. Cyclic photophosphorylation. It is the normal process of photophosphorylation in which the electron expelled by the excited photocenter does not return to it. Non-cyclic photophosphorylation is carried out in collaboration of both photosystems I and II. Electron released during photolysis of water is picked up by photocenter of PS2 called P680. The same is extruded out when the photocenter absorbs light energy Hv. The extruded electron has an energy equivalent to 23 kilocalories per mole. It passes through a series of electron carriers Q, PQ, cytochrome complex and plastocyanin. While passing over cytochrome complex, the electron loses sufficient energy for the synthesis of ATP. The electron is handed over to photocenter P700 of PSI by plastocyanin. P700 extrudes the electron after absorbing light energy. The electron passes through X, Phase center A, ferredoxin, and NADP reductase which combines it with NADP+ for becoming reduced H+ released during photolysis to form NADPH. ATP synthesis is not direct. The energy released by electron is actually used for pumping H+ ions across the thylakoid membrane. It creates a proton gradient. The gradient triggers the coupling factor to signal 1E size ATP from ADP and inorganic phosphate. Oxygen evolution. The oxygen that is evolved during photosynthesis comes from water and it is part of the photoact mediated by PSII. The light energy trapped by this system excites P690 and 2 electrons are ejected. The energy is used to remove 2 electrons from the hydrogen of the water and boost them to a higher energy level. At this point, the molecular oxygen which escapes from the photosynthetic system is formed. The electrons pass through the carriers plastoquinone PQ, cytochrome B559, cytochrome F, plastocyanin and finally end up in P700 to take it back to the ground state. Thus in photosystem 2, the electrons that brings the excited chlorophyll molecule to the ground state comes from photolysis of water. Another aspect of oxygen evolution during photosynthesis is its relationship to the presence of certain ions in the medium such as Cl, Mn2+ and bicarbonate. 2H2OO2 plus 4H plus 4E. Both photosystems working in union produce, for every 2 turns, 2 molecules of NADPH2, 3 ATPs, and a molecule of oxygen from 2 water molecules. Some herbicides block electron flow. The use of herbicides to kill unwanted plants is widespread in modern agriculture. Many different classes of herbicides have been developed, and they act by blocking amino acid, carotenoid, or lipid biosynthesis or by disrupting cell division. Other herbicides, such as DCMU, dichlorophenyldimethylurea, and paraquat, block photosynthetic electron flow. DCMU is also known as diurin. Paraquat has acquired public notoriety because of its use on marijuana crops. Herbicides, DCMU among them, act by blocking electron flow at the quinone acceptors of photosystem 2, by competing for the binding site of plastoquinone that is normally occupied by QB. Other herbicides, such as paraquat, act by accepting electrons from the early acceptors of photosystem I and then reacting with oxygen to form superoxide, O2, a species that is very damaging to chloroplast components, especially lipids. Carotenoids serve as photoprotective agents. In addition to their role as accessory pigments, carotenoids play an essential role in photoprotection. The photosynthetic membrane can easily be damaged by the large amounts of energy absorbed by the pigments if this energy cannot be stored by photochemistry. This is why a protection mechanism is needed. The photoprotection mechanism can be thought of as a safety valve, venting excess energy before it can damage the organism. The energy stored in chlorophylls in the excited state is rapidly dissipated by excitation transfer or photochemistry. The excited state is said to be quenched. Institute for Advanced Studies, JADPR 5. Institute for Advanced Studies, JADPR 6. If the excited state of chlorophyll is not rapidly quenched by excitation transfer or photochemistry, it can react with molecular oxygen to form an excited state of oxygen known as singlet oxygen, 1O2*. The extremely reactive singlet oxygen goes on to react with and damage many cellular components, especially lipids. Carotenoids exert their photoprotective action by rapidly quenching the excited state of chlorophyll. The excited state of carotenoids does not have sufficient energy to form singlet oxygen, so it decays back to its ground state while losing its energy as heat. Mutant organisms that lack carotenoids cannot live in the presence of both light and molecular oxygen, a rather difficult situation for an O2 evolving photosynthetic organism. For non-O2 evolving photosynthetic bacteria, mutants that lack carotenoids can be maintained under laboratory conditions if oxygen is excluded from the growth medium. Recently carotenoids were found to play a role in non-photochemical quenching, which is a second protective and regulatory mechanism. Some xanthophylls also participate in energy dissipation. Non-photochemical quenching, a major process regulating the delivery of excitation energy to the reaction center, can be thought of as a